So we've got a packed room. Welcome to CTRB Contributed Oral Presentations about DNA and genetic resources. So welcome to everyone in the room and everyone that is online. I'm not sure how many are online, but welcome everyone. We'd like to thank our Diamond sponsors for all of their support and for all the other smaller sponsors that have allowed us to all be here today. So thank you to them. So if you are online, please place your questions in the Zoom chat, or even if you're in the room and want to ask a question but just not put your hand up, if there's not enough time, put it into Zoom. I'll put it into the Oxford Abstracts chat features, please. We'll be monitoring that. The sessions are going online fairly quickly, so yeah, if you miss anything or you want to review, they will be online soon, but please be patient with our volunteers that are madly uploading imagery. So for our speakers, you have 10 minutes, the green light, and then you have another couple of minutes, and then when it's red, I'll come up and tackle you and take you down, all right? So all good. So first up, we have planning for the future. Is it time to subsample types? And we've got Chris Milensky. Come on up.
Thank you very much. I always worry when your talk is assigned to the smallest room on the last day that there might not be an interest, so it's nice to see a packed house here. As stewards, our goal is to preserve specimens and objects for current and future research. The use of natural history collections as a genetic resource is the primary request type that we are receiving now in the bird section at the Natural History Museum. And so I'm primarily going to be talking about dry collections, and you'll see in this photo the nice arrangement of a bunch of our different dry collections, and it's a very nicely organized, and for traditional taxonomists, you might see lots of things you want to put your hands on, but what a lot of our research are seeing when they look at this photo is DNA and the idea of using these specimens for DNA research. And so the technological advances in DNA have allowed people to get whole genomes now from 100-plus-year-old specimens. And so while these specimens are stable in their nice dry storage cabinets, the DNA is not necessarily stable as it is continuing to degrade over time. And so I'm going to talk a little bit about storage conditions. And so this chart here, graph, shows specimens that were obtained of birds at the Smithsonian Institution by decade. And there's a lot to unpack here, but basically if you look at this and you look, the majority of our specimens were collected prior to 1970 and prior to storage of tissue samples in freezers. And so what that means is that those specimens have been under a variety of storage conditions and were not really what you would call optimal storage conditions until after 1960, when they were moved into the current space that we have with proper HVAC and humidity control and specimen cabinets and that sort of thing. So these specimens have been through a lot, and so that has impacted the DNA for sure. But that being said, we still have specimens that are being used in research using toe pads, removal of toe pads. These are some passenger pigeons, and you can see the toe pad label in the middle when that sample was removed in the 90s, and with a result of whole genome of passenger pigeon that was published in Science in 2017 with Martha on the cover. Martha, the last passenger pigeon, she was not sampled for that study, but this is what our molecular folks, what they're thinking about is how to use these specimens for research. These samples do require sometimes very special techniques in ultra clean rooms with Tyvek suits and masks and all that sort of stuff. And the reason is because you have this curve here that shows how much the DNA is lost really, really rapidly. And so these specimens are, while the DNA is still there, it's barely there. And the farthest graph on the right is actually something interesting that we discovered as I was researching for this talk, is that counter to what you might think, larger specimens are worse for DNA from older specimens. So the largest specimens produce lower quality DNA than small specimens, and that's what that line is. The small, all those little dots up on the top upper left, were DNA extractions from smaller specimens and quality, and then coming down as the quality goes down as the specimen gets larger. And so why is that? That was an interesting question. And what we determined was that it was related to how quickly the specimen dries. And so if it dries slowly, the DNA is worse. So what we decided to do is we're going to go ahead and try to stop that DNA degradation by taking samples and putting them into our biorepository. This is a very large facility with over 6 million tube capacity, and is not anywhere near full, so there's space. And so we said, well, maybe we should think about freezing some of our stuff. But going back just one little step here, the way the birds are prepared, the quality of the DNA can vary greatly. There was a paper published that said that DNA has a half-life of 521 years, but that was at stable conditions for that entire life of that sample. And if you think about samples that were in less than ideal conditions, which a lot of our specimens were, the half-life is going to be a lot less than that. Or if you think about some of the frozen, things they find in the tundra that are 30,000 years old and still have good DNA. So it's really a variable number, but the way that the specimens are preserved in toe pads, if the specimen had the toes opened and dried more quickly than ones that did not, and they also tend to be a little bit less greasy when you do that. And so those are the same species, two different methods. One person opened the toes and one person did not. And there's a question about which is going to have better quality DNA, the one that was opened or not. And I suspect it's the one that was opened up and dried more quickly. So just a little overview of value of specimens. If you just do a quick Google Scholar search on using museum specimens for genetics, you find dozens and dozens of papers over the last 10 years or so that talks about how museum collections are this wonderful resource for specimens and DNA. And some of those examples, you have skins, skeletons, eggs, fluids, your basic DNA, microbiomes, parasites, disease, all of these kinds of requests we have received used for use of our specimens, older specimens. eDNA, of course, being environmental DNA, is hugely popular now, and people are thinking about all kinds of different ways that they can get DNA other than the actual host from a specimen. We've recently just had someone who wanted to take tape and actually get samples off of bills and feet of raptors to see if they could study the diet of these raptors that were, and I think it was not a very good idea, and I think we denied that request but people are thinking about it and thinking about all kinds of different ways to use specimens. So for this project, we said, okay, we're going to sample our types. We're going to cut toe pads off. How are we going to do that? What's the process we're going to use? And so we said, first thing we need to do is document. So we were able to get internal Smithsonian grant to do photography of all of our type samples. 5 photos per specimen, front, back, side, and front and back of the labels. And we hired an outside photographer contractor to do all that work, and it's wonderful. These are all now online. You can go onto our website or onto any of the data aggregators, and you should be able to see all of our types photographed. The biggest debate we had was sampling protocols. We have a committee and a lot of opinions. And so we ended up coming up with something that more or less everybody was okay with. But there was a lot of things to think about, and especially when you had type series, if you had 10 co-types of the same thing collected at the same place, do you really need to take 10 toe pads? And so we decided not to, and only do about half in those situations. But we had to come up with some basic protocols and trying to keep clean using fresh scalpel blade with every specimen, of course, and things like that. So this is what it ended up looking like. We have a published book of types, Dagnan's types. We also have all of those in our database. So and they're all in a type collection. So we just simply clicked types and downloaded that and sorted it all out. And we had this nice little book that we could use while we were doing the sampling with our tubes, pre-labeled tubes with our biorepository number on them with a barcode. And then we started cutting. And so that's what a little toe pad looks like when it was taken off the specimen. One of the things, if you read quickly through all those, you notice that we were cutting these in half with the idea that somebody's going to use half in the future to actually do genetics and get a genome or something from these specimens. Ideally, you would do that right now, but that would be really expensive. So we're hoping that the cost comes down so that we can do whole genomes from all of our types. But cutting them now, if you've ever tried to cut something really hard in half, like a grain of rice, pew, 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 they go flying all over the place. So we decided we would do that under controlled circumstances rather than having somebody in the biorepository trying to cut something in half out of the tube later. So we did that in advance. This is just quick. This is what our database looks like. GenSample 1, we call this tissues and parts, mixed tissue, and then topad and verbatim. Emo is emo. However, one of the big debates was whether or not you really need to be doing this. In 40, 50 years from now, is topads what people are going to want to do genetics? And so already, this is not a bumblebee tickle torture device or something. This is somebody actually just taking a swab and getting DNA from that bumblebee without doing anything destructive to it. And so that was my biggest concern as collection manager was like, is this really going to be the answer in the future? And how long do you have to store something in the freezer before it becomes better than just taking off the specimen again? But we did it. And so we now have all of those samples that will be there and waiting for someone to do genomics. But then it also came up the question of storage and freezer space. And this paper just came out 2 weeks ago saying that if you are worried about your freezers and storage of freezers, you can just store things on the moon. It's nice and cold and you don't have to worry about freezers failing. But anyway, that is the end of my talk. Hi.
Thank you for the talk. Awesome. I love DNA. I've been working with DNA for, 50 years. How about you can came anyway, right? No,
the online people, yeah.
Okay, sorry. I'm not going to repeat what I just said. But anyway, thank you for the presentation. So I have maybe comments, if I can.
Yeah.
So the graph about larger animals and lower quality of DNA. So then we're talking about quality of DNA, you mean fragmentation, right?
Yeah.
So, and also the larger animals, do you mean also bones? Because you also can extract from bones. So bigger animals have more dense bones.
I'm primarily talking about topads. So
Okay.
there have been 2 papers published now that have said that topads from larger animals did not work as well, and they suggested potentially using skin instead.
Okay, so you mean just birds?
Yeah, I'm
because
talking
I thought about bigger
Yeah, I'm thinking about
whatnot.
so it's about how quickly it dries.
It dries, yeah.
And so and for birds, it's the topads. Yeah.
And another one, you talked about cutting the tissues, and yes, they fly.
Yeah,
and it's
yeah.
really hard to cut, and it's very messy, it's very annoying, it's time consuming.
Yeah.
But my approach was that actually I soak them in a lysis buffer, so they get a bit softer.
Yeah.
So then I can take them out and cut very easily. Have you tried it? No?
No, because we're trying to sample 4,000, and we're doing it in our dry collection space and not in the lab space.
I see.
So we weren't thinking about soaking them first. One of the protocol ideas we had was actually some of the protocols for bird topads is to put them in alcohol.
It dries. if you put in alcohol.
Yeah.
And it's also even harder to cut.
And we decided not to do that, but but we did not think of, just in terms of amount of time it would take, I think we didn't think about trying to soak it first before we cut it. But what we did was, because we had a clean scalpel blade, foil, we just put the toe pad back into the foil and cut it inside the foil so that it wouldn't fly away.
That's a smart technique as well, but it's really hard. It's like cutting a plastic.
Yeah, no, they're hard. They're hard.
Yeah. Thanks. Thanks a lot.
Okay.
Thank you.
All right.
Right, next up we have Ricky Lee, who is going to be talking about bringing invisible genetic samples into focus. Thank you. Take it away.
Thank you. Kia ora and hai tai. My name is Ricky Lee Erickson. I'm the collection manager of land vertebrates and the tissue collection at Tamaki Pangahira Auckland War Memorial Museum, who also has natural history collections, and my pronouns are she, her. Sorry.
Tēnā koutou katoa, ko Ricky Lee Erickson, tōku ingoa. I am the collection manager of the Land Vertebrates and Tissue Archive at Auckland War Memorial Museum, Tamaki.
I think it's the video, not the slides. Oh. I can turn the volume down, and then I can just talk as it plays. Does that work? Yep. Okay, I'll just pause it. Okay, so I'm going to talk about the Marine Threatened Species Tissue Archive, which was funded by the Department of Conservation, and our aim was to initiate an archival tissue sample storage system for protected fishes and marine reptile bycatch with their associated metadata, and this would include nationally important collections and samples tenuously held within universities. So tissue collections that are not lodged within museums or institutions with collection management and databases are at risk of dissociation and physical deterioration. We wanted to provide sampling kits to fishery observers and participating commercial fisheries. We wanted our sample storage and curation to follow accepted international standards and include appropriate cultural controls. And we wanted to ensure that these samples were accessible. So the Marine Protected Species Archive includes sea turtles, sea snakes, kraits, and some sharks, fish, and ray and invertebrate species. And the main threat of to these species are commercial and recreational fishes. Other threats include ship strikes, habitat degradation, and pollution. And with warming waters, more turtles than ever before are reaching New Zealand, which is something that's different in New Zealand, at least. And it is difficult and expensive to get genetic samples for these species. A lot of them live in really deep waters or really far offshore. So we want to ensure that bycatch fatalities are not wasted, and we want to ensure that these samples are accessible to researchers. Okay. We also wanted to develop the museum's existing tissue collection as part of this project, which have collections from the marine, land vertebrates, entomology and botany departments. And these are all in various stages of development, depending on the research activities of both former and current curators and research associates. We needed to standardise the storage of these samples, and we didn't really have a means to import tissue-related data onto our museum database, which is Vernon CMS. And our data was not meeting the Global Genome Biodiversity Network, GGBN standards, which is like the Darwin Core for genetic samples. We accessioned our samples as parts of a voucher specimen, which can be problematic. So this is one of the problems of accessioning the tissues as part specimens. It's difficult to search on how many samples do we actually have, and we have these messy data downloads or exports from Vernon. So any field that has, you can say which part of the tissue it is, whether it's current location or storage method, it concatenates all of the data into one field, which makes it really difficult to work with. unfortunately, we couldn't add any user-defined fields in Vernon. So we had to, and like you saw in Chris's fields, EMU has all these amazing windows with lots of fields for genetic information. So we had to repurpose existing Vernon fields. And Vernon was really helpful to help us find fields to store this information. And we also collaborated with other museums that use Vernon CMS that have genetic collections, such as Otago Museum and Queensland Museum. We mapped the GGBN core fields to Vernon fields, and we developed a new tissue window to be used in conjunction with standard departmental window. So you'd have your botany window open, and you'd also have your tissue window open, so you could access the fields if you're working with collections, tissue collections. This is an example of what the new tissue window looks like. It can be quite bare, but can be quite full depending on the samples. And one of the key things is that we accessioned each tissue sample as its own record, and we linked these records together using a related record feature, which means if you search for the voucher or for this tissue, both records will come up. So it's quite user-friendly in that sense. We also standardised our storage for the tissue archive. So we had on the left tubs of tissues in various storage containers, mostly though much too big for what was required. And we moved these into standard 1.5 ml cryovials, and the location of each tissue position within each box is noted. So it's really easy for us to access the tissues now, whereas before it took us a bit longer, and it's obviously bad to let specimens defrost while you're trying to find them. We also needed to ensure that we could document any cultural permissions required for tissue samples. So we used a window outwards, inward rights reproduction use, which was already in Vernon. And we were able to, this also allows us to import permit information, which is part of a GGBN standard. This is an example of some frog toes that were given to us by Ngāti Kuata, a Māori group. And these are endangered frogs, taonga species, sacred species. So any use of these toe pads, toes, sorry, not pads, just toes, whole toes, we need to get consent from Ngāti Kuata, and we need to inform them of the results of this research. And this is something that is going to become more and more common, and not just in New Zealand, but other parts of the world. So what worked well in the project? The Department of Conservation is the governing agency in New Zealand, and they were driving and funding the project. They have the top authority on tissue samples, so they can direct researchers and institutions to transfer their samples to us. Also, having a dedicated collection manager for the archive is essential to develop the collection and ensure that the correct procedures and protocols are observed. And also a dedicated project collection technician who is still working right now in New Zealand at migrating the samples from the storage, and also migrating their associated data on Vernon. Some of the challenges. So we're dealing with poor historical tissue curation, resulting in data loss. One of the major acquisitions we've had was a collection of over 2,000 turtle samples, not 2,000 individuals, but 2,000 tissue samples. And unfortunately, we were only able to accept about 600 of them because a lot of the data had been dissociated just using Excel over the space of 10 to 15 years, and also the physical labelling of the tissues. So that's very common, unfortunately. We have a lot of following up leads and researchers for tissue samples, harassing them, but so that takes up a lot of time, especially when you're working on other collections. Like my other job is the land vertebrates collection, so that can be difficult. Getting those GenBank IDs or other repository IDs off of researchers doesn't happen that often, and so I could only easily find 14 records on GenBank from Auckland Museum, and I know we've done countless tissue grants to researchers. We had a pretty low uptake with fisheries. They were hesitant to provide location data. They want to protect their fishing grounds. We do restrict all of our records for this protected marine species archive, so only certain things are viewable, but they're obviously still a bit hesitant. And also their procedure is, if they get something on the ship, if it could still be alive throw it back overboard immediately in case it could survive. And they're not biologists, so it can be hard to tell, especially with these, deep sea sharks that do have very low metabolisms and can be hard to tell. And Vernon CMS is probably not the best database for natural history collections. I worked with EMU previously, but it's still working okay for us, and that's what the museum is sticking with. One of the downsides with linked records is if you update your classification or any other information, you need to ensure that you are updating both records. So this is what the archive looks like at the moment, mostly turtles and white sharks, about 700 individuals with lots of duplicates. And we're going to maximise our scientific value of genetic samples, now visible on Collections Online and GBIF. We can report tracking sample use and cultural permissions. We've modified the database and we're growing the collection. A couple of really cool examples of the collection so far. Leatherback turtles sample sent to Australian Museum. We're going to figure out where these turtles are coming from in New Zealand, and also sending some fish samples to Otago University for a molecular library so we can use techniques like eDNA in the future. Kia ora and Nife Deberu.
Does anyone have any questions in the room? How are we doing online? Do we have anyone online? If you could have whatever CMS you could possibly have, what would you choose?
I've only used EMU, but I'm interested in Specify. That looks good too, so I'd have to play around with that a little bit.
Excellent. Well, we've got one more question. I don't think this will reach, so.
Just very briefly, you had mentioned that you're working with the Indigenous communities as well and providing the results. Do they have infrastructure and data management, things like that, in place for the community so they have equal opportunity to look at the data?
Yeah, one good thing about one of the great things, I'm Australian working in New Zealand, is that the development of iwi or communities of Maori is much greater, and they're easily contactable. We're constantly consulting with them. It's a very two-way sort of street, which is really good. We have really great relationships, and they have really developed organisational structures, committees, working groups. So they're able to disperse that information very easily. And
All right. So how are we doing for time? We're okay? We're good to go? All right. All right, so now we have Cynthia. Come on up. She's talking about the review of practices for preserving animal genetic resources. Is there enough room there?
Do I use the mouse? Or can I use the keyboard?
You can use the mouse. The other.
Oh. There you go. All right. I will move this keyboard out of the way. Okay. All right. Hello, everyone. My name is Cynthia, and I recently started as the Manager of Genetic Resources at the Museum of Comparative Zoology at Harvard. Today, I will review some of the more practical side of tissue preservation and curation, especially as we receive specimens from the field, or tissues from the field, and transition it to the museum. And at the very end, I'll share some of my thoughts on future directions for the collection. All right, so the main principle of preservation methods is to slow down or stop the degradation of genetic materials in your tissues. And the method that you choose can significantly impact its potential uses in the future. So depending on the research goals, as well as the transportation requirements of your samples, certain methods may be better suited than others. So some of the questions that you should ask yourself include, what are the types of biomolecule types that you need? What is your budget? Do your samples need to be kept cold? and what are some of the legal requirements with using, shipping, and transporting your particular taxon and the preservative method that you're using. So I'm just going to go through 4 of the primary methods that we see in the literature. So the first one is 95% ethanol. Ethanol is great in that it is very accessible. It's relatively inexpensive compared to some other methods, and it preserves DNA for long periods of time at room temperature or below. The downside is that it is flammable, which means that if you're planning to transport it either in your carry-on or on a plane, or if you're shipping it to another institution, you do need to abide by International Air Transport Association Special Provision A180 requirements, which you can find online. The other downside is that not all markers are ethanol resistant. For anyone who has ever tried to write with Sharpie on anything, you spill some like cleaning supplies on it, it disappears, which in the case of our samples is disastrous if some of your samples leak. The second most common preservation method that that comes across my desk is DMSO, also known as dimethyl sulfoxide. These often come in solutions of that comes with EDTA as well as sodium chloride. You'll see it in the literature as Sutin solution or DESS. Although ready-made solutions are not commercially available for DMSO, you can prepare it very easily and cheaply in the lab. The great thing is that it's not considered a shipping hazard, so that's one pro above the ethanol. But the downside is that over time, DMSO will precipitate in your vial. So what ends up happening is that your little piece of tissue at the bottom of the vial becomes encrusted with salt, and that salt, when you transfer it into extraction methods, may interfere with your ability to actually get DNA out of your sample. All right, the third one is RNA later. RNA later is great in that it can preserve RNA. It, however, is temperature sensitive, so there are certain guarantees as to how long RNA is good for in your sample. And it is susceptible, it is also very sensitive to the ratio of the liquid that you're using and the tissue amount that you put into the vial. The literature also recommends that you dice the tissue up very finely so that the RNA later can permeate through all of your tissues. The big downside for RNA later is that it is very expensive. When I looked up the price last week, 100 milliliters is $174 US, which is mind-boggling for most researchers who are in the field who have very limited funding. The other thing to be aware of is that because RNA later is preserving RNA, any potential viruses that are that exist in your tissue could be restricted in terms of your ability to transport it across borders. So for example, in the US, US Fish and Wildlife has restrictions on bird tissues coming into the country because of avian flu. Lastly, we have freezing in liquid nitrogen or flash freezing. This is considered the gold standard of preservation methods because at minus 180 degrees Celsius, all biological and chemical activities essentially stop. If you're going into the field, one way to bring liquid nitrogen with you is the use of a dry shipper here. Essentially, how it works is that you pour liquid nitrogen through the top, it gets absorbed by this blue layer here, and then the evaporation of liquid to gas cools down the container. So if you're in a country or a part of a country where you have access to industrial gas production, you can use this dry shipper to keep your samples cold for a few days or up to a couple of weeks, depending on what equipment you have. However, this means that if you're in the field for longer than a couple of weeks, you need to drive back to the liquid nitrogen production area and top off your dry chipper on a regular basis, which can be challenging depending on where you are. The other things to be aware of are, because the temperature that you achieve through liquid nitrogen is so cold, proper PPE is strongly recommended, especially a face visor, as samples that have been submerged in liquid nitrogen can sometimes explode when you take it out of your dry shipper. There have also been some studies that suggest that for samples that are not just flash frozen, things that have been put into another buffer or preservative, that these ones that have been soaked in a liquid can sometimes perform better than flash frozen tissues when you're in extractions, because it kind of gives it some protection as the tissues thaw. All right, so here's just a quick summary of what I just presented to you. If you want this as a PDF, or if you have any questions, feel free to email me. I'm happy to share it with you. All right, so on to some of the more practical considerations. So in the MVZ, we typically rehouse our tissues into cryovials and apply barcodes for easy tracking in our database. But we found that not all inks are very friendly with our workflow. As I mentioned earlier, some inks are not resistant to ethanol, and you end up with illegible labels on your vials. And we've also found that some inks are not stable under our barcode sticker. So here you can see that this ink has just kind of diffused in between the cryo vial and the sticker, whereas other ones like Nice Day or Smart Plus tend to do really well. And as I mentioned also earlier, cracked vials is another problem that we've been experiencing right now, but I don't have the good answer for how to get around that yet. All right, so thus far, we found that markers that are designed for marking microscope slides tend to do really well with barcodes. If you don't have barcode on hand, it's quite resistant to ethanol. However, it can be scratched off, but that's not really an issue for us if we can get the barcodes on it relatively quickly. As for cracked vials, the general practice is to not pack it chock-a-block full of whatever buffer or tissue you're sampling, and also to apply the barcode before freezing, because that's much easier. And that also can, the barcode sticker can also hold the vial together so that when you're having to transfer it to another tube, you can just pluck your frozen material out without it leaking everywhere. All right, so in our database called MCZ-base, right now it is very voucher-centric, with tissues tracked as parts of a voucher specimen. Some of the data is stored as controlled vocabulary, such as dry or frozen, and whatever could be frozen in -80 or liquid nitrogen. But a lot of it is stored in an open text field right now, such as the tissue type and the time the tissue was taken after euthanizing an animal. This information on timing is actually really useful for RNA studies, but it's sometimes overlooked by external researchers who are interested in our specimens, as it's not intuitively searchable within the database right now. We also regularly accession small organisms, such as parasites, into the cryogenic collection, and we're able to add relationships in the database like that. All right. So two of the things that I've been asking myself as I sort of settle into my position is, one, how do we increase the accessibility of our genetic samples in our museum database? And for anyone who attended the GGBN session earlier this morning, that has given me a lot of food for thought for how to make our tissues more accessible. And the second question I've been thinking about is how to future-proof our resources by anticipating future methods and uses. I feel like this is especially pertinent right now with the increasing limitations to going into the field, whether it be permitting or access to other countries. Should we, as museum staff, be more forward in collaborating with field researchers to maximize the quality of the samples that they're collecting before either environments disappear or collecting becomes impossible due to permitting? So in conclusion, careful consideration of preservation methods is needed when maximizing the future use of tissue collections, and collaborating with a lot of other people who use our specimens will be key to establishing standards to optimally preserve, store, and use samples. So thank you for your attention. I'd love to hear more about what other museums are doing in your collections, and I'm happy to answer any questions you might have. All right, thank you.
Thanks, Cynthia. That's a great overview. Does anyone have any questions? Oh, we've got another. Got a hand up.
Yeah, sorry, I don't want to seek too much of attention, but DNA is sort of my big part of my life.
Yeah.
So I have a question. As you are talking about taking tissue samples for DNA in the field, I don't know, maybe I missed it somehow, but how do you connect species, the sample, right, with your tissue sample? Do you take some photos? And if you do, some measurements maybe as well? How do you connect in your database? Because that I kind of missed.
So all of our tissues are added as parts of a specimen record. So each specimen record usually has a voucher specimen in it. So we usually can add supplementary information like photos, permits, collector, measurements. Like
I see.
all of the data is ultimately hinging on that specimen catalog number.
I see.
Yeah.
Thank you.
Mm-hmm.
Hi, thank you for that overview. I had a question about the barcoding labels and whether or not you needed to use a specific kind to withstand -80 temperatures or not. We're trying to set up our own cryopreservation setup, and we're not sure if we need to, if we can just use printer labels or if you need to have a special freezer-resistant type. So,
yeah. So in my experience, we have special ordered stickers from a company based in New Hampshire, and they are rated to withstand cryo temperatures. I have seen people use paper labels, but they start to fall apart when you freeze thaw samples, and after a while it just disintegrates.
perfect. Thank you.
Yeah.
I have a comment that kind of goes with that question as well. Something a little cheaper than ordering the specific freezer ones is Brady labels makes a cryo tube. I've been testing it for a couple of years now, and I have the Brady M211, which is a Bluetooth printer to your phone. So you can literally text the printer in the field the label, and that label is freezer-proof, alcohol-proof. I've been soaking them in alcohol for a couple of years and freezing them in -80 liquid nitrogen, and the labels are holding up really well to that, which is slightly cheaper for the average user than the specialty barcoding things that our cryo facility uses.
Mm-hmm.
And it works great in the field.
Okay, yeah, I'd love to learn more about that.
Yeah, I can pop it in the chat. We've been testing it for a little while, and it's so far been really good and a cheaper option for the normal person. You know, the printer's only $150.
Yeah, awesome, thank you. Yeah. Yeah, sorry.
I'm interested in the PPE advice about using face masks due to cracking vials. Couldn't cracking vials be avoided by only using dry shipper with nitrogen vapor as opposed to submerging in a liquid? Is there a reason for this choice?
Yeah. The problem stems from user error. I'm not going to call out any specific demographics, but sometimes you can overfill your dry shipper to be able to stay in the field longer. So when samples get thrown in, it just kind of swims in liquid nitrogen for a while, and that's not something that I have a lot of control over, but I do recommend to people to not submerge in liquid nitrogen. Yeah. Yeah.
Well, thank you, Cynthia.
All right, thank you.
All right, great talks. All right, so now we have Emily. I will leave it to you to pronounce your very special last name. And she's going to talk about the key to bringing DNA collections to the next level.
Hi, everyone. My name is Emily Veltjen, and I'm going to talk about the key to bringing your DNA connections to the next level. So first off, you saw DiSSCo there in the title. So for those who don't know what DiSSCo stands for, this is a distributed system of scientific collections. I redirect you to the website. That's very informative. But in short, the whole concept is about digitally unifying all natural science collections in Europe and also providing user services that will allow, will facilitate reuse. DiSSCo Flanders, you could see it as a concept that's nested within DiSSCo Europe, right? So also a distributed system of scientific collections, but then in Flanders. Flanders is the northern region of Belgium, in green on the slide. But we also use the term DiSSCo Flanders to, when we talk about the project, a project that we have, or the consortium. And so when talking about the project, we are talking about all these different institutes. So we have the Flemish project partners, but also associated partners, and also federal and Walloon associated partners. So it's a little bit bigger than Flanders. And each of these institutes, they have their own natural science collections. So there's a big variety here, but we're talking about DNA samples, so DNA specimens here today. So in the project of DiSSCo Flanders, we had, or there was a need identified to share best practices about DNA collections. And for that, we instated the DiSSCo Flanders DNA Collections Working Group, which is a group of about 50 members, which were allowed to be as passive or active as they wanted. And in total, we will have held 10 meetings. So the next meeting is still in October, November. And the whole concept was to share best practices, inspire one another, and also to brainstorm together. We have a simple shared Google folder where we put in information and an email address that allows quick exchange of information. And well, originally the plan was to also write a deliverable on best practices for DNA collection management. But of course, once we started sharing and inspiring and brainstorming, we saw that, okay, we already have the ISBER best practices. And then with the Synthesis Plus project, hola, a lot of information also became available. For example, the Biodiversity Biobanking Handbook that you saw maybe in the earlier session, and also the Policies Handbook. So, well, we decided not to write another deliverable on best practices because this information is great. But there was still a big question in our group, like, okay, how are we gonna put theory into practice, right? So this is all great information, but where do we start? How do we do this? And also some of, for example, the ISBER best practices is more general compared to the more specialized DNA collections. So we still had this question, like, okay, how can a DNA collection be matured as efficiently as possible? Also taking into account the collection's diversity in context and size. And so when talking about context, in our group, it's very variable. First of all, available means, right? I think that's a problem that everybody has. And then also the scope of the DNA collection, geographical scope, taxonomical scope, but also eDNA, aDNA, ancient DNA, environmental DNA. And then the genetic lab, so the organization of the genetic lab, that's sometimes associated with the DNA collection. Is it centralized, decentralized? How does it work? How many people are allowed to work in that lab? Type of institute, so our working group, it's not only natural history musea, for example, it's also governmental institutes and also universities that are in this working group. And then, of course, people is always very important. So who is managing the collection and who is supplying DNA samples, DNA specimens to the collection. So we brainstormed, and then we came up with two outputs. So the first is the challenges, and the second output is the key. And the key is more or less the tool that I will talk about. But first, the challenges. So it's a simple list of 23 challenges. We explicitly state them to find solutions for the key, so for the tool, and also just to generally spark debate. It's now in its first version. It's really the vision of our group, but we will put it out there, and then we hope that more people will join in the discussion. An example is, for example, number 8, select or customize a collection management system to meet the needs of DNA collections. So there's 22 more, right? And that brings me to the key, so the second output that we created. It's a tool, it's a yes or no questions. There are 7 yes or no questions, similar to a taxonomical key. So you go through them, and when you say yes, you can go to the next question, and then no, you have to undertake some kind of action. So these are the 7 questions. I shortened them a lot, so the real questions are longer. And the first 4 questions really help you think about your strategy, like of your DNA collection. So they comprise like looking at internal stakeholders, looking at your institutional scope, the collection scope, and also like where do we stand right now? What is our current workflow? And for that, a great resource is this book. It's about all natural science collections of Huxley and colleagues. And then the real work begins, actually. So the first 4 are pretty easy and straightforward to answer. But number 5 gives you this chart, which is a maturation chart, we call it. It has categories, 11 of them, and then 3 levels. And then each category can have a level 1, a level 2, and a level 3 goal. For example, the legal compliance category, the level 1 goal would be that you inform at least the suppliers of the regulatory frameworks and that you provide them with a central logging space, workspace for logging. The level 2 would then be, for example, that you instate 5 legal checkpoints. And the level 3 goal would be that you could state that your DNA collection is completely legally held, as far as you know. Right, so that's just one example for one category, but there are 10 more. And so what you have to do in step number 5, or question number 5, is, for example, for this hypothetical My DNA collection, is that you plot yourself on this maturation chart, so you see where you're starting. And in this example, for example, 3 categories are not, none of the goals were met, which is perfectly fine. And then we say, okay, you start at level 0, and you take it from there. And then you have checked off question number 5, so you establish your starting level, and then you can continue to 6, which could take even longer than number 5. So the goal is to reach level 3. So you do this in a stepwise manner, as fast or as slow as you can with your available resources. So the first step for those 3 that were on level 0 is to go to level 1, and then you try to move to level 2. So now the collection is at level 2, and this is the end point, right? So you try to comply with the goals of level 3. And then you could say, okay, my DNA collection is fully matured. And that leaves only step number 7, that's to instate a reevaluation strategy, so that you think about, okay, at what time interval will we redo this exercise to check if we're still compliant? The context can change of your DNA collection, or best practices can also change, of course. The key comes with guidance chapters. So for the 7 questions and the 11 categories, there is extra information that will guide you to all the relevant literature that we found, but maybe there's more. And each time these guidance chapters are in the same format, so they reformulate the question, explain it a little bit more, give you the literature, stress the importance, and then at the end, it also states the experiences, so the user experiences with that specific category or question. So at this point, it's only the user experience of our working group that's in there. But this way you can also learn from each other in a more passive format. Good. So the key We think it will help us a lot because it allows a more of standardized, holistic way of maturing your DNA collection. It provides like a helicopter view with a maturation chart, but it also breaks it down in manageable steps, so you can focus on one goal at a time. It also allows you to set priorities because, of course, the categories that are lagging behind, you have to address first, and it's highly relevant or specialized for DNA collections. You can use a key standalone or within a community, which is what we would like to do. So the ambitions, so we're going to put this information out there. It's not out yet, but it will be in a post-publication peer review in Rio, so others can join in. With the working group, we want to test, optimize, and update the key. We want to do this in two ways. So a key manager, so somebody will convene the different DNA collections and help them with mapping on the tool. And on the other type of session would be that the different DNA collections or DNA collection managers share their experiences or their ambitions for the next half a year, for example. We think the tool has some possibilities. You could recycle it for other types of natural science collections. Of course, we can upscale it because now it's only the working group that provided input and the literature. And of course, it can be reformatted because at this point it's a simple document, but it could be a more interactive tool as well, for example, with a GitHub website. All right, that was it. If you would like more information, you can find me after or send me an email. And then I also want to thank the co-authors, all the participants of the working group, also the invited speakers, and our funding. And you, of course, for listening as well. Thank you.
Thanks, Emily. Does anyone have a question in the room? We don't have anyone online as yet. You never know. Fantastic. Thank you so very much, Emily. I should come up with a question or two for people, I suppose. We do have just a little bit to wait, just so that people can come in and leave as they feel free. But in the meantime, if you'd like to come up and get ready, we have Catherine. Stephan? I apologise. Yes. Stephan is going to be talking about opportunities and limitations of the FAIR principles for collections of plant genetic resources. But we'll just hold on just one more minute, if you don't mind.
Sure.
Just in case someone wants to run over here and listen to you.
Just get me the sign to start. Okay. Hello, everybody. I would like to speak about a currently running project that we are involved in. And our interest here in special is the opportunities and limitations of FAIR principles when applying them to collections of plant genetic resources. As everybody of you know, in life sciences, we have to cope with an increasing or exponential growth of research data over the last couple of years. You can see this from the example on the right-hand side of this slide. There's a graph showing the increase of sequence data from NCBI and GenBank, and you see that they are already counting in zettabytes, and this also holds true for other data domains. Nevertheless, research data is very important, should be available to everyone in the world at all times, of course. It bears a high potential, It helps to reduce duplication of work, can increase the efficiency of research, enables more comprehensive research results, automated processing. It helps to develop new research questions based on existing knowledge. However, the main challenge is to manage those massive amounts of data in a way that it allows to generate new insights, valuable insights. And of course, the so-called FAIR principles can help here to achieve those goals. They help to, or can help to, make research and research results in general more transparent, more efficient. They promote collaboration, and they can help to break down silos in research and increase sustainability in general. I think I don't need to go too much into detail. FAIR stands for findable, accessible, interoperable, and reusable. That means a dataset needs a unique and persistent identifier to be able to find this dataset. It should be accessible and retrievable by using a standardized protocol. Consistent vocabularies should be used. There should be links to metadata to make this data interoperable. And important part is the reusability. That means especially what is often lacking in the area of plant genetic resources. We need rich descriptions of metadata describing how an experiment was conducted, when, who, by whom, and how was the setup, and so on. And as I said, we are working on plant genetic resources. Around the world, there are about 1,800 collections of plant genetic resources, depending on how you count. And amongst them, about 600 are located in Europe and in some neighboring countries. And in total, we are speaking about 5.8 million accessions that are being maintained in gene banks and other collections under ex situ conditions, so outside of the natural habitat. And of course, data, good data, high quality data is just crucial for maintaining the material, for sharing the material with researchers, with breeders, and especially for exploiting the material, to make it usable for researchers, for breeders, and also for private persons. And our special focus is on Europe, as we are located in Europe, and we think it matters for European PGR data to try to reach fair compliance because it allows a holistic data management. It helps to improve collaborations, to adapt conservation strategies, to optimize the utilization of plant genetic resources in general, and also to improve policy development. And when we are talking about PGR data, there are different data domains that have to be taken into account. For example, ex situ passport data, in situ data. Very important for PGR collections are phenotypic observations, so-called characterization and evaluation data, increasingly genomic data and also image data, and many other data domains. And so far, most of the data sets in the plant genetic resources community do not fully comply with FAIR principles. There are still lots of limitations and obstacles, but nevertheless, there are some ways forward. And I would like to show you some examples. We still have to cope with non-standardized collection identifiers. There's a high fragmentation of data sets across institutions, very heterogeneous data sets. And it's unfortunately rather unlikely that we will ever experience standardized collection identifiers. But a way forward is to use additional unique identifiers. And for that reason, FAO recommends to use permanent unique identifiers in the form of DOIs. They are increasingly accepted and used by gene banks, but there is still a great potential for improvement. And in Europe, we have DOIs so far for approximately 20% of the gene bank holdings. The same number holds true for the whole world. In case of genetic data, there are biosample IDs that are being issued by the 3 global players, NCBI, GenBank, EBI, DDBJ. They can be linked with DOIs, which is a very good improvement. And what is still very necessary is to apply DOIs on a broader scale also for existing phenotypic data sets. The accessibility to PGR data is still often hampered by restrictions. There are different storage systems, different technical infrastructures, and a special challenge is the sustainability of those infrastructures. There are some international aggregator systems in place, such as DiSSCo, Genesis, ViewViews. They are really nice, but they need to be expanded. The networking between those systems need to be improved. And a special need is to develop trusted repositories, especially for phenotypic data, for plant genetic resources collections. This is one of our focuses to work towards this, and to consistently make people aware to submit their project data to public repositories to have them available for the future. Also, the interoperability is hampered by semantic issues. There are different format standards that are being used. And here there are still a lot of things to do with regard to harmonization of terminologies and standards. There are approaches for semantic standardization, but they are not yet fully developed and not yet effective. This especially holds true for phenotypic data. There are ontologies in place, of course, and as a computer scientist, I really like ontologies, but the way they are being used in plant genetic resources communities does not really work properly. So we think that restrictions might have to be accepted here. Yeah. And also the reusability of PGR data is hampered by missing metadata, incomprehensive documentation, incomplete data, incomplete standardization. And here it's very important to consequently make people aware, push them to use existing approaches for better description. For example, MyAP, minimum information for plant phenotyping experiments, is in place, can be used. And it's very important to push people to also apply this to project data, also to legacy data. We are working in one of the projects doing exactly this, and to make people aware to early involve also data stewards. Why am I talking about this? In Europe, we have the vision to develop a fair, compliant European PGR information system. The coordination nucleus will be DiSSCo, the European Search Catalog for Plant Genetic Resources, which is already in place, having at the moment 430 different collections providing data. This system will be extended by more harmonized vocabularies, additional data structures, increased interoperability. And at the moment, we are involved in a project called Progress, promoting a plant genetic resource community for Europe. This is a pan-European approach coordinated by colleagues from Italy. And the aim of this project is to develop a concept, a framework, and a governance for a plant genetic resources research infrastructure for Europe. And of course, we are seeking synergies with other European research infrastructures. The project is in contact with other infrastructures, such as Emphasis, for example. And our central aim here is to develop the existing DiSSCo system into an integrated European PGR information system by adding missing sources, connecting additional domains, promoting standards, protocols, providing lots of service and training. And by this, we think that we can make a good contribution towards making European PGR data more fair in the next couple of years. Thank you.
Thank you, Stephan. we do have one comment online for you. It is, go find it. It's from Andy Bentley. And he says, DOIs are not fair. They cost money and there are still far too many collections that will not be able to afford minting DOIs for all of their objects. We need a sustainability model to fund DOIs for other parts of the world if it's to be a global solution. Do you have a comment?
The decision for using DOIs was made by the FAO. There was a task force that was initiated in 2015. They discussed different types of identifiers like ARCs, life science identifiers, DOIs. And in the end, the majority decided to use DOIs. They are technically not the best solution, I completely agree, but they are widely accepted. And the International Treaty for Plant Genetic Resources of Food and Agriculture, which is located at FAO, they implemented infrastructure that allows all gene banks around the world to mint DOIs for free.
Thank you. Does anyone else have a question? We have a little bit of time. You've stunned them all into silence. No? Okay. Well, thank you very much. Very interesting. All right. We'll just wait a little bit because we've got a few minutes so we remain on schedule. Everyone is way too efficient. Takeru, would you like to come up and get yourself comfortable maybe in the meantime? So I've got the mouse and the pointer, and if you want to use the keyboard instead. Yeah? Okay. While we're waiting, there is a follow-up question from Andy. Could you provide more information about the free model for minting DOIs, please? Could you provide that more information about the free minting of DOIs that you mentioned? Or you could answer offline as well, if you like. Yeah, perfect. All right. So next up, we have Takeru, who is going to talk about aspects of NCBI GenBank as a biodiversity information resource. Take it away.
Hi everyone, my name is Takeru Nakazato from Tokyo, Japan. The title of my talk is the aspect of NCBI GenBank as a biodiversity information resource. I'm biomatician. This photo is from a biohackathon. Biohackathon in Japan is just finished, and biohackathon in Europe will held in November. Many bioinformaticians are working on data integration and standardization of DNA sequence-related data. I'm interested in linking sequence data and biodiversity data. This is an example of GenBank nucleotide data, as you know. Initially, as I was searching for eDNA sequences on GenBank nucleotide, and I realized that there are various qualifiers related to biodiversity labeled pink. So I downloaded all the NCBI GenBank nucleotide data and considered the situation and possibility of linking it to biodiversity information. This list is a summary of qualifiers related to biodiversity. INSDC prepared qualifiers for specimen IDs, sampling date and place, and identification. This year, qualifier for geographic information was changed from country to geolog name to align with biosample specification. In Darwin Core, location information is separated into country, state, province, county, and municipality. But INCD's design, geographic data is written in one column. This graph shows the percentage of records that each qualifier is assigned. I surveyed about 7 million insect records. The organism qualifier is 100% because it is a required item that describes the organism name. And the DBXDF qualifier is also 100% because taxonomy IDs corresponding to organism name are described here as external database IDs. Interestingly, about 35% of insect nucleotide data are assigned with specimen IDs. And collection date and collected by almost the same situation. This graph shows the torrents of records with specimen voucher data. I surveyed the situation of insects, fish, and flowering plants. I'm sorry, this is a real number of records, but currently INS in recent years, specimens ID have been consistently assigned to 30% to 40% of records. Currently, INSCC requires spatio-temporal metadata, that is geolog name and collection date. This may be an excuse that compared with Nagoya Protocol, but as a result, it means that the amount of sequence data with biodiversity information will increase. Type specimens are important for biodiversity research. Type information is designed to be described in the type material section. But in some sequence records, such information is described in the node or specimen voucher section. The values listed in type material are defined as a controlled vocabulary. Please search for INSDC type material for detail. This is one example of data relationships in GenBank, INSDC, and BOLD. These three data are duplicated because origin is one sample. But these data differ data source, so three records exist, makes sense. I'm not sure we should merge or not, but at least I think it would be useful to have these data linked together. Yeah, exactly. Each database provides each ID. So in this slide, many IDs occurred. That makes the situation complicated, I think. In that context of data linkage, some specimen IDs referred in GenBank nucleotide have a link to Arctos database. It is an example for a bird specimen. I have not yet comprehensively examined the status of such link, but I love insects, so I search insect sequence, but most of insect sequence assigned with specimen ID, but there are no link. So it may be a lucky situation, but yeah, I need to survey comprehensively, I think. Specimen ID matching is required to create a link. NCBI maintains collection code in their biocollections database. And I have heard about the activity of GR cycle. Yeah, so problem is the same ID matching problem, I think. And in near future, AI technology will assist such matching task, I believe. Yeah, I was inspired at this conference to increase the link between GenBank and GenBank data and occurrence data and specimen data such as GBIF. I hope the link will increase and become more useful in the future. Thank you very much.
Thank you, Takeru. That's fantastic. Does anyone have any questions for Takeru? Yeah, come on up.
Hi, thanks a lot for your talk. I'm Joanna, I work at the eDNA, and it's not so much as a question, it's more like a comment and some updates related with some things that you have been presenting. So we are fully aware of this situation, and I agree, this is complicated, especially when we're trying to match legacy data that we have at INSTC with specimen information. We have been making efforts towards improving that. We had, for example, in the Elixir Hackathon last year, we had a project that was trying to map GGBN references to data in INSTC, for example, and also to tell you that NCBI biocollections and GR source cycle are actually in contact, and they are trying also to at least keep each other's identifiers. So that it's more easily to map. So I guess it's very good to raise awareness on this and for everyone to try to move forward on these linkings. Thank you.
Thank you very much.
Oh my. Okay, Andy Bentley has written a little essay for us. Thanks, Andy. It's great to have you online. Would it not be more efficient to link to the specimen record in the original CMS or an aggregator like GBIF and pull it into the GenBank record to ensure that the specimen metadata is up to date? This is the essence of integration rather than duplication, as in the extended specimen. INSDC also needs to start using Darwin core terms to make it easier to express this data. There are so many cases of incorrect information associated with sequences from my tissue collection, and I do not have access to change these. Only the submitter can do that, and I have had no luck in getting them to do it. It's an ongoing challenge for probably most of us in the room. We're trying to update data. Yeah. Do you want to comment, or you're all good?
I'm satisfied.
Yep. That's all right.
Okay. Yeah. So regarding that last issue on updating, it is not possible to update because it is user submitter, but we do have at EBI a system for curating. So if you're interested, you can get in touch and we can send you some more information on that, and you can submit curations towards that. We do use Darwin Core for some fields, but users don't always comply. And there is, and on another note, GBIF has a clustering system, I'm not sure if you're aware, where they try to cluster samples that come from different places. Not always the information is available for that. And I agree that it's good to have everything linked in between. Thank you.
All right. Nope, there is still nothing on the, well, except for Andy. Yay, Andy. Thank you, Takeru. That's fantastic.
Thank you very much.
Yes. And we have the very lucky last, Rachel. We've got a comment from Andy. Make yourself feel at home. Yeah. Clustering relies on the metadata being available in both sources, which compounds the problem. Yes. We're getting a nodding head in the audience. Yes, that everyone agrees. All right. We just have a little bit of a wait, but not too long, just to make sure that everyone who wants to come and see your fabulous presentation can get in the room. Actually, we've got a 5-minute wait.
Okay.
Sorry.
Should we tell a story? Or?
Yeah, do a little dance, a little interpretive action. Introduce yourself, what are your likes and hobbies. See, someone came in. It was worth the wait. Welcome. I feel like I'm turning into Deb Paul somehow. That's not a good thing. Deb's great. I don't want to replicate Deb. Is there any questions in the room for any previous speakers? Anything jumped into your head in the last 5 minutes? No? Oh, yeah, come on up, Alan.
Sorry, I was standing outside. I've got a question for Cynthia. So for plant DNA, we use silica gel a lot, and we were wondering about long-term storage, having put in silica gel at the first stage. And some places, apparently you can lyophilize it as well and put it in filter papers and hold it. Has anybody had any experience of that? Is it any good, or should we just keep it in silica gel?
I unfortunately don't have any experience with plant material, but I am curious to learn more about what you guys do to preserve your materials, because sometimes I have worked with bird blood preserved on filter paper, but the long-term preservation and quality, I have no idea like how to just move forward with that and improve it. So, but we can borrow something from Plant World.
So the nice thing about silica gel is you can take it in the field, bit of a tea bag, bit of leaf, bag. Occasionally you're caught in a hotel room with white powder all over the floor, but apart from that, there's fairly low risk. As long as you're sensible, masks, gloves. We do use things like DNA, RNA later for living material, but usually in the gardens. I'm not entirely sure what we do in the field if you're after that stuff. But yeah, it's we've stopped routinely pulling, extracting DNA because it's a lot of work, and often researchers want to do it themselves anyway with the primers they've got. So our principal store is plant tissue, held in drying cupboards and Tupperware boxes, but that's bulky. So we're wondering, is there a long-term solution? The nice thing about the DNA is you can more or less store it in a herbarium if you've got the temperature and stuff like that, so it's very stable. But just wondering if there's any long-term thoughts about freeze drying, or is it worth it? If anybody knows out there, let us know.
All right, Rachel. I don't know how to pronounce that first word. I should have asked you earlier. So I'll let you introduce yourself.
Okay.
Thank you.
So, hello everyone. My name is Rachel Haderlé. I'm a PhD student at the ISIAB lab at the Dina Marine Station of the Muséum National d'Histoire Naturelle in France. So today I'll be discussing environmental DNA, focusing specifically on taxonomic assignment in vertebrates. So to start with some background, our understanding of biodiversity is still incomplete. In order to plan and monitor effective conservation strategies, it is crucial to conduct biodiversity assessments. Organisms leave DNA traces in their environment, such as feather or saliva. Environmental DNA, or eDNA, refers to DNA that can be extracted from an environmental sample without prior isolation of the organisms. eDNA metabarcoding enables the genetic simultaneous characterization of several taxa within a single environmental sample using short DNA sequences. eDNA metabarcoding is particularly useful to study hard-to-sample ecosystems, to detect rare or cryptic taxa in a non-invasive way, and to conduct naturalist inventories. eDNA metabarcoding can be used in a variety of contexts, from the open sea to coastal environments, to study anthropized sites or more preserved sites such as MPAs. So there are three major issues dealing with eDNA metabarcoding. So particular care must be taken in order to avoid potential false positives or false negatives inherent in the study of trace DNA. A particular attention must be paid to the taxonomic assignment of DNA sequences, which requires strong skills in molecular taxonomy. And finally, a special effort has to be made in order to standardize and share eDNA data in biodiversity databases. Here, I will focus on the second point. So in my case, after conducting a sampling plan, seawater is collected by continuous filtration along a specific transect, and then samples are analyzed using metabarcoding principles in a specific dedicated lab. And it generates millions of reads. Then bioinformatics processing is a crucial stage, and I won't go into much detail here, but it enables to group reads into motus for molecular operational taxonomic units, based on their molecular similarity. Basically, it reduces millions of reads to a few hundred motus. And then these motus are compared to reference databases in order to assign them to taxa. And it is at this taxonomic assignment stage that there are major differences between protocols. And currently, there is no consensus about which protocol is the most appropriate. So my study focuses on this point of taxonomic assignment. And in fact, many authors reduce the challenges of taxonomic assignment to incomplete reference databases. But there are other major issues to consider, such as low interspecific variation or ecological features which are often not considered. And all these problems highlight the need to involve taxonomic experts, so both classical taxonomic experts and classical and molecular ones, into this taxonomic assignment process. Automated taxonomic assignment can be prone to errors. So, for example, this motu that we have detected in Brittany along the coast was assigned to the Atlantic sailfish. So quite unexpected species for the area. So we checked with the GenBank and we got these results here. And you can see that on the first line, we got one sequence of Atlantic sailfish, yes, but only one. And all the other ones, they belong to the Angrolidae family, the anchovies. And this family is quite distant from the Histiophoridae family. So in fact, this first sequence is a complete mitochondrial genome. And we know that errors can occur on short sequences in such read assembly. So here, it might be an error of automated taxonomic assignment. And this error could be due to an error in the database of reference. Here is another example of a motu that we have detected this time in Guadeloupe, so in the French Caribbean, which was assigned to the Indo-Pacific bottlenose dolphins. So we checked with the GenBank, and we got high similarity values with several species of delphininae, which are locally distributed. So one goal of my PhD is to refine these taxonomic approaches. That's why today I am presenting a taxonomic assignment protocol for vertebrate modules designed to verify their automated assignment and to check their consistency. So our protocol was created using an iterative process based on an eDNA dataset that we have just published this year in a data paper. And it is a 4-steps process, which is presented here as a flowchart, coded in Mermaid using ISO standards for the symbols. So step 1 involves comparing our module to the GenBank using the BLAST tool. And if we have a barcoding gap, all the most similar species are listed up to this gap. Otherwise, the top 2% similar species are listed, and it generates our mole list of the most molecularly similar species. Then step 2 involves listing the species of each genus in the mole list using taxonomic reference databases, and it produces the taxo list. Then step 3 determines the geographical distribution of the species using resources like FishBase, and it produces the geolist. Then the goal is to create the MTG list, which selects species present in both the mole and the geolist. And it is at this stage that we can identify if we have species from the geolist which are not included in the MTG list. And if so, we can check if their barcode sequence is present or absent from the GenBank. And if it is absent, we have identified that this species barcode was not in the GenBank and that we had a lack in the reference database. So finally, we can create our last list, which is the hypo one for hypothetical taxa, which is basically the MTG list and eventually lacking species that we have identified, which are then particularly flagged. And you can then perform your taxonomic assignment to the most precise taxonomic rank from this hypo list. So our protocol is currently the only one existing to assist taxonomists in their taxonomic assignment of eDNA metabarcoding results. It allows us to go beyond standard taxonomic assignments protocols as it generates a list of hypothetical taxa. It can identify if we have potential species which are lacking from the reference database and incorporates geographical distribution criteria. So using this protocol, I have already studied an MPA in Guadeloupe where we have identified 3 species of marine mammals and 66 species of fish. In Brittany, in an estuary, we have highlighted a strong estuarine gradient thanks to eDNA vertebrates metabarcoding results. And currently, we are working on other eDNA datasets, including one on marine wine farms. So our protocol produces good results, but it is quite time-consuming and repetitive, trust me. So in order to facilitate its reuse, we are also trying to implement it as an automated tool. So that's why I'm currently working on an OurShiny app, which is still in progress. The idea here is to import your module list and your BLAST results. And then for each species, you can check if it is locally distributed or not, and also manually add a missing species from the GenBank, and then it generates the hypo list. So there are still a lot of improvements to be made here. I'm a biologist and not a developer, so I'm also here seeking for advices, particularly on good practices in order to advance on this project and to make the code for this tool as open as possible. And finally, this project is just one aspect of my PhD. I'm also doing lab work. I'm trying to develop bioindicator metrics applied to eDNA results. And I mentioned a data paper earlier, so we're also trying to standardize and share our eDNA data on biodiversity databases using the Darwin Core and Mixes interoperability standards. And yes, all these efforts are centered around the broader objective of studying mobile marine fauna using eDNA. And that concludes my presentation. I thank you all for your attention.
What a fantastic PhD project.
Thank you.
Does anyone have any questions? Oh, would you mind passing this back?
It's more like to everyone. If anyone noticed a correlation between these error sequences in the GenBank and when they were uploaded. I was thinking that maybe if they were uploaded 20, 30 years ago, the method were not that great as they are now. Maybe that's what produces the error. So has anyone ever checked that or thought about it? Or is just me kind of dreaming it? Thank you.
So I haven't particularly noticed that, but I understand your point of view, so maybe it will be great to check that kind of stuff, yeah.
Also, just a comment of if you are trying to develop this out, the Untraws Direct set of tools allows you to use an API to retrieve BLAST results, and so that might be something to look into as a good way to automate this process. Just as a
yes. So it's also a goal after when everything will work perfectly. So not now, but yes, after it will be great to not to add manually each BLAST result. Yeah.
All right. Well, thank you so very much for your talk. And thank you to all of our speakers today. Thank you. Thank you to Daphne for helping out up the front here and to our magical team up the back. Yeah, well done. Have a chat about DNA while you're all in the room. Yes, and thank you. Enjoy lunch.
That's okay. I'm lucky.